Good afternoon, class. Today we're starting a new unit, Unit 4, Chapter 3, Part A. So we're going to be looking at cells. The human body is built on, it is made up of several trillion tiny units called cells. And in fact, cells are the smallest entities that can perform all the basic life functions that we discussed in Chapter 1. Yet, unlike a single-celled organism such as an amoeba, a cell in the human body must depend on its neighbors, and the actions of one cell can affect all others. Our cells have not only to survive, but also to work together in relative harmony, coordinating their activities. Each cell has a particular role to play, and in its own way, each cell helps the body to maintain homeostasis. Section 3.1, cells, the smallest living units. So the cell theory states that a cell is the structural and functional unit of life. How well the entire organism functions depends on individual and combined activities of all of its cells, and structure and function are related. Biochemical functions of cells are dictated by the shape of the cell and specific subcellular structures. And the continuity of life has cellular basis. In fact, all cells come from the division of pre-existing cells. Cells have a variety of forms and functions. There are over 250 different types of human cells, and these types differ in the size, the shape, and subcellular components, and these differences will lead to differences in functions. Figure 3.1, cell diversity, shows different cells in the human body that perform different functions. So in A, we see cells that connect body parts like the fibroblast cells. Epithelial cells will form linings all over our body. And the erythrocytes, also known as red blood cells, that transport gases, oxygen and CO2. Then cells that move organs and body parts like skeletal muscle cell and smooth muscle cell. They have this long shape. In C, we see a cell that stores nutrients, an adipose cell or a fat cell. In D, we see a cell that is involved in immunity, a macrophage. Macro is big, phage is it's a big eater. It will attack bacteria and viruses. And E is a cell that is responsible for gathering information and controls body functions, so a neuron. And F shows a sperm cell, which is a cell of reproduction. Notice it's adapted for swimming because it's got a flagellum. Although cells come in different shapes and sizes, they all have some common structures and functions. So a generalized cell is made up of three basic parts. One, a plasma membrane. They all have this flexible outer boundary. Second component is a cytoplasm, which is intracellular fluid that contains the organelles. And thirdly, all cells have a nucleus, which contains the DNA, which is the control center of the cell. Figure 3.2 is the structure of the generalized cell. Notice in bold, if we start on the right, we can see a nucleus, the nucleus that contains the DNA. Here it's shown in the form of chromatin. A plasma membrane that surrounds the entire cell, and a cytoplasm, which is found within the cell, and it contains all of the organelles, which we're going to learn about. Let's look at the extracellular materials. Extracellular means these are substances found outside the cells. So classes of extracellular materials include extracellular fluids or body fluids, such as an interstitial fluid. So cells are bathed in this fluid. So that's the fluid surrounding the cells, as opposed to intracellular, which is the fluid within the cells, right? That there's a difference between interstitial and intracellular. There's blood plasma, which is the fluid component of blood. And cerebral spinal fluid, or CSF, is the fluid surrounding the nervous system organs. Also, there are cellular secretions such as saliva and mucus, and extracellular matrix, which is the substance that acts as glue to hold cells together. Part 1, plasma membrane. So the plasma membrane acts as an active barrier that separates the intracellular fluid compartment and the extracellular fluid compartment. It plays a very important role in cellular activity by controlling what enters and what leaves the cell. So it's selectively permeable. It allows certain molecules to come in and not others. And it's also referred to as the cell membrane. This figure shows the different fluid compartments. So the fluid compartment within cells is called the intracellular fluid, and the extracellular fluid shows interstitial fluid, which is that fluid surrounding the cells, and plasma, which is also known as intravascular fluid. Section 3.2, structure of plasma membrane. So the plasma membrane is made up of membrane lipids, mainly the phospholipids that we learned about in the previous chapter. And these will form a flexible lipid bilayer. Bilayer means that there are two layers of these phospholipids. Also, a component of the plasma membrane are specialized membrane proteins that flow through this fluid membrane. So the membrane is referred to as fluid because it's not a rigid structure. And the result is that these membrane proteins will change. You won't find them in a fixed position. So that's why it's called the plasma membrane is referred to as a fluid mosaic pattern. It's fluid. Mosaic means that it's made up of different components, not just phospholipids, but also proteins and also other components like sugars, which we'll talk about. So yes, surface sugars form what is known as the glycocalyx. So they're found on the outside of the membrane. And the membrane, there's also membrane structures that help to hold cells together through cell junctions. So this is a portion of the plasma membrane, and it's called the fluid mosaic model. There was another model before that was proposed for the composition of this membrane, but this is the one that's accepted. So notice it's made up of mostly phospholipids, and it's also got those large proteins embedded in the membrane. And remember, we talked about the oligosaccharides, or those sugars that are found on the outside of the membrane. There's also some embedded cholesterol you can see that is associated with the animal membranes. Figure 3.1, the plasma membrane. So on the left, the functions of the plasma membrane. So it acts as a physical barrier, separating, we said, the extracellular fluid compartment from the intracellular fluid compartment. It's a selectively permeable membrane because it will allow certain molecules to enter and others to exit. It acts as also for communication. So there are plasma membrane proteins that interact with specific chemical messengers and will relay messages to the cell interior, and acts as cell recognition. There are cell surface carbohydrates that we talked about in the previous chapter called oligosaccharides that allow cells from the, for example, immune cells to recognize that it's part of it's part of self. We say it's part of a person's makeup of cells. So here also we can see the phospholipid bilayer in the figure, in purple, those large proteins embedded in the membrane, and the oligosaccharides, those carbohydrates that form the glycocalyx. The second part of figure 3.1, the plasma membrane. And I do apologize for the poor quality of the image. I could not get it bigger than this. Anyway, so the three components of the plasma membrane are the lipids, the proteins, and the carbohydrates. For lipids, the main lipid is the phospholipids. These form the basic structure of the membrane. Remember, the polar hydrophilic heads will interact with the intracellular fluid inside the cell and the extracellular fluid outside. And then you have the hydrophobic nonpolar tails that will orient themselves towards each other, keeping water away. Another lipid is cholesterol, which helps stiffen the membrane. And then we have proteins, and proteins are determine what functions the membrane can perform. So it could be transport, communication. So you can have integral proteins, which are integrated within the membrane. They're embedded in the membrane. You also have peripheral proteins, which are not embedded. They're found on the outside. And then we have the third component, the carbohydrates, which act as identity molecules, we said, and they form the glycocalyx. So those carbohydrates are short chains of linked monosaccharides, and the oligosaccharides. There's several of those glucose monomers joined together. And they're attached to proteins. So if they're attached to proteins, they're called glycoproteins. And if they're attached to the phospholipid, to a phospholipid, then they're called glycolipids. But both of them together, glycoproteins plus the glycolipids, are the glycocalyx. This table simplifies the components of the plasma membrane, also giving their location. So phospholipids, we said, are the main fabric of the membrane, are the main component of the membrane. Cholesterol is tucked between the hydrophobic tails of the membrane phospholipids. And then you have two types of proteins, the integral proteins and peripheral proteins. The integral proteins are the ones that are embedded in the phospholipid bilayer. They may or may not extend through both layers. Peripheral proteins are on the inner or outer surface of the phospholipid bilayer, but they're not embedded in its hydrophobic core. And then we have those carbohydrates, which make up the glycocalyx, and they're attached to proteins or lipids on the extracellular side of the membrane. So if they're attached to proteins, they're called glycoproteins, and if they're attached to lipids, they're called glycolipids. Here's an animation of the membrane structure. You're going to have to look in your PowerPoint and play it in your PowerPoint, but I like this figure because it shows the orientation of those phospholipids that make up the membrane. So notice it's a bilayer. Bilayer means that it's two layers of phospholipids, and the yellow part is the hydrophilic heads. Remember, it's made up of the glycerol component and the phosphate group component of the phospholipid, which we discussed in the previous chapter. So those will face or naturally orient themselves towards the extracellular fluid and the cytoplasm, which is fluid and it's watery, and they like water, right? Because they're charged. And then the hydrophobic tails will position themselves towards each other because they're hydrophobic, right? And they don't like water, don't like to be in contact with water, because remember, the tails are made up of mostly carbons and hydrogens. They're hydrocarbons. Okay, so membrane lipids, a little bit more detail. It's nice, some repetition. It helps cement this information into our brains. Okay, so lipid bilayer is made up of 75% phospholipids. And we said they consist of two parts, the phosphate heads that are polar, they're charged, so they are hydrophilic or water-loving. And the fatty acid tails, that component is nonpolar, there's no charge, because remember, it's mostly carbons and hydrogens, so they are hydrophobic or water-hating. Then there's 5% is the glycolipids. These are lipids with sugar groups on the outer membrane surface. Remember the oligosaccharides that are bonded to lipids. And 20% of the membrane is 20% of the lipid bilayer is made up of cholesterol, which helps increase membrane stability. On the left, so this section 3.2, structure of plasma membrane. On the left, we can see the icon for a phospholipid. So it's made up of two major components: the polar hydrophilic head, which includes the phosphate group, which is negatively charged, if you remember, and so that's why it's polar, and the nonpolar hydrophobic tails, which is made up of the fatty acid component, the hydrocarbon component. And if you place a bunch of those in a watery solution, in an aqueous solution, they will spontaneously orient themselves, as seen in the figure on the right, where the polar heads face the water inside and outside the cell, and the nonpolar tails will hide from the water. So membrane proteins, these will allow cell communication with the environment. They make up about half the mass of plasma membrane. Most have specialized membrane functions. Some will float freely and some are tethered to intracellular structures. There are two types: integral proteins and peripheral proteins. Let's look at the function of integral proteins. So these are firmly inserted into the membrane. Most are transmembrane proteins, which means that they span the entire membrane from one side to the other. They have both hydrophobic and hydrophilic regions. So the hydrophobic areas will interact with the lipid tails, the lipid hydrocarbon tails, and the hydrophilic areas will interact with the water. So what that means is that you would have hydrophobic amino acids that interact with the lipid tails and hydrophilic amino acids on those proteins, which are part of the proteins that interact with the water component. They function as transport proteins, such as channels and carriers. They can function as enzymes or receptors. Peripheral proteins are bound to the inner or outer surface of the membrane and are easily separated from it. In other words, they stick to the inner or outer surfaces of the membranes. Integral proteins greatly outnumber peripheral proteins. So they're loosely attached to integral proteins. They can include filaments on the intracellular surface. They're used for plasma membrane support, and they can function as enzymes, motor proteins for shape change during cell division and muscle contraction, and for cell-to-cell connections. Just one final note on enzymes. So enzymes in the cell membranes can be integral or peripheral proteins. And these enzymes catalyze reactions in the extracellular fluid or in the cytosol, depending on the location of the protein and its active site. For example, dipeptides are broken down into amino acids by enzymes on the exposed membranes of cells that line the intestinal tract. Figure 3.3, membrane proteins perform many tasks. So let's zoom in and examine each one of these tasks. Figure 3.3a shows transport. So there are two examples in the figure. So on the left, the protein spans the membrane. So it's an integral protein and will provide a hydrophilic channel across the membrane that is selective for a particular solute. On the right, there's some transport proteins that will hydrolyze ATP and use it as an energy source to actively pump substances across the membrane. Another function of membrane proteins is acting as receptors for signal transduction. So in this figure, we can see a membrane protein has that is exposed to the outside of the cell may have a special binding site that fits the shape of a specific chemical messenger, which could be a hormone. And when the hormone binds to that binding site, it's going to, it may cause a conformational change in the three-dimensional shape of the protein, and it initiates a chain of chemical reactions in the cell. For example, when insulin binds to the receptor of a cell, and it's going to cause changes in that cell in terms of, let's say, uptake of glucose from the blood. Figure 3.3c, we see another function of membrane proteins as enzymes. So in the figure, there's a set of enzymes that have their active site exposed to substances in the adjacent solution. And often you can have a team of several enzymes in a membrane that may catalyze sequential steps of a metabolic pathway. Figure 3.3d, membrane proteins acting as for cell-to-cell recognition. So some glycoproteins, remember these are examples of oligosaccharides that are bonded to proteins that make up the Glycocalyx, serve as identification tags that are specifically recognized by other cells. Figure 3.3e, some membrane proteins serve as attachment to the cytoskeleton and extracellular matrix. Elements of the cytoskeleton, which is the cell's internal framework, and the extracellular matrix, fibers and other substances outside the cell, may anchor to membrane proteins, helps maintain the cell shape, fixes the location of certain membrane proteins, and also plays a role in cell movement. Figure 3.3f shows the role of membrane proteins in cell-to-cell joining. Membrane proteins of adjacent cells may be hooked together in various kinds of intercellular junctions. Some membrane proteins, called cell adhesion molecules or CAMs, of this group provide temporary binding sites that guide cell migration and other cell-to-cell interactions. So the glycocalyx is the third component of the cell membrane and consists of sugars or carbohydrates, or specifically oligosaccharides, sticking out of the cell surface. Some sugars are attached to lipids, and they're called glycolipids, and some to proteins, and those would be called glycoproteins. Every cell type has different patterns of this sugar coating, which functions as specific biological markers for cell-to-cell recognition and allows immune system to recognize self versus non-self. I explained this concept a little bit in the previous chapter. So, in terms of their role in recognition, the glycoproteins and glycolipids are recognized as normal or abnormal by cells involved with the immune response. And the characteristics of the glycocalyx are genetically determined. For example, the presence or absence of membrane glycolipids on circulating red blood cells will determine your blood type. Remember, we talked about A, B, or O. And the body's immune system can recognize its own membrane glycoproteins and glycolipids as self rather than as foreign or non-self. And this recognition system keeps your immune system from attacking your blood cells while still enabling it to recognize and destroy foreign blood cells should they appear in the bloodstream. Other functions of the glycocalyx are lubrication and protection. The glycoproteins and glycolipids form a viscous layer that lubricates and protects the cell membrane. Another important function of the glycocalyx is anchoring and locomotion. Because the components are sticky, the glycocalyx can help anchor the cell in place. It also participates in the locomotion of specialized cells.
All cells have an outer membrane, called a plasma membrane, which acts as a boundary between the outside and inside of a cell. Cell membranes are primarily made of phospholipids, which are large lipid molecules, similar to triglycerides. Like triglycerides, they contain glycerol and fatty acids, but they differ from triglycerides in that they have a phosphate group, rather than a fatty acid, attached to the third carbon of glycerol. The phosphate head of the molecule is hydrophilic, or water-loving. This means that it interacts well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not interact well with water. Because of these unique properties, when placed in a liquid environment, phospholipids tend to arrange themselves into a phospholipid bilayer, so that only the hydrophilic heads interact with a watery environment, and the hydrophobic tails crowd inward, away from the water. The plasma membrane of a cell also contains other molecules that aid in its function. There are a number of different proteins embedded in a membrane, and they serve a variety of functions. Some are involved in the transport of substances across the membrane, while others help in cell-to-cell communication. Short chains of sugars are attached to the outer surface of some of these proteins, forming glycoproteins, which serve as recognition signals to other cells. Cholesterol is present in the membrane as well. Cholesterol lends support to the membrane and regulates fluidity, giving the membrane the general consistency of olive oil. The phospholipids, proteins, glycoproteins, and cholesterol are all present in the membrane as a flexible pattern, commonly referred to as the fluid mosaic model. Together, these components of the cell membrane allow the cell to interact with its external environment.
Clinical Homeostatic Imbalance 3.1. The glycocalyx of some cancer cells can change so rapidly that the immune system cannot recognize the cell as being damaged. And the mutated cell that escapes recognition by the immune system is not destroyed, and so it is able to replicate unchecked. Next section is cell junctions. So some cells are free, which means they're not bound to any other cells, like epithelial cells. So there are examples of free cells, like blood cells, like red blood cells and white blood cells, and sperm cells. But most cells are bound together to form tissues and organs. There are three ways cells can be bound to each other, either through tight junctions, desmosomes, or gap junctions. So tight junctions, these are integral proteins on adjacent cells that fuse to form an impermeable junction that encircles the whole cell. They prevent fluids and most molecules from moving in between cells. And where do you think they might be most useful in the body? Figure 3.4a, cell junctions. The first type is the tight junction. So these are impermeable junctions. They form continuous seals around the cell and prevent molecules from passing between cells. So a location for this in the body would be the intestines to prevent leakage into the extracellular matrix. The second type of cell junction are desmosomes. These are rivet-like cell junctions formed when linker proteins or cadherins of neighboring cells interlock like the teeth of a zipper. The linker protein is anchored to its cell through thickened button-like areas on the inside of the plasma membrane called plaques. Keratin filaments connect the plaques intercellularly for added anchoring strength. Desmosomes allow give between cells, reducing the possibility of tearing under tension. Where might these be useful in the body? Figure 3.4b, desmosomes. So these are anchoring junctions. They bind adjacent cells together like molecular Velcro, and they help keep cells from tearing apart. So where would you find these cells in the body? So they're found in tissue that experience intense mechanical stretch, such as cardiac muscle or bladder tissue, gastrointestinal mucosa, and epithelia. The third type of cell junction are the gap junctions. These are transmembrane proteins, also known as connexons, that form tunnels that allow small molecules to pass from cell to cell. They're used to spread ions, simple sugars, or other small molecules between cells. And they also allow electrical signals to be passed quickly from one cell to the next cell. So you can find them in cardiac and smooth muscle cell, like the walls of the heart, to make sure that the atria will contract in unison and the ventricles will contract at the same time. Figure 3.4c, we can see gap junctions. They're communicating junctions. They allow ions and small molecules to pass from cell to cell, and are particularly important in heart cells and embryonic cells.
When cells come in close contact with one another, they develop specialized intracellular junctions that help the cells communicate and connect. There are three major types of intracellular junctions in animal cells: tight junctions, anchoring junctions, and gap junctions. Tight junctions seal adjacent cells together tightly to one another, sort of like a zipper, and form a barrier that cannot easily be crossed. Cells connected by tight junctions serve as barriers in the body, sealing off cavities and preventing leaks. Anchoring junctions connect neighboring cells in particular spots, but do not seal the two cells together. In anchoring junctions, internal plaques attached to the cytoskeleton are joined by intercellular filaments. This creates a sheet of cells that is strong but also flexible. Anchoring junctions form in organs where tissues must stretch, such as the stomach or bladder. Gap junctions connect neighboring cells with a protein channel that allows for communication and also for the transfer of small molecules and ions. Gap junctions are important in heart muscle and smooth muscle because they allow for the flow of ions that let these cells contract as a unit. All of these junctions are important for forming strong connections with one another, establishing communication, and preventing the passage of unwanted materials.
Section 3.2, structure of plasma membrane. So many substances must constantly move across the plasma membrane, and some molecules pass through easily while others do not. So we say that the plasma membrane is selectively permeable, allowing only certain molecules to cross. There are two ways that substances cross the plasma membrane, either passively through passive transport where no energy is required, or through active transport where energy in the form of ATP is required.
A semi-permeable membrane, also called a selectively permeable membrane, is a membrane that allows certain molecules or ions to pass through it while blocking others. One example of a semi-permeable membrane is a phospholipid bilayer, a group of phospholipids consisting of a phosphate head and two fatty acid tails, arranged into a double layer, with the hydrophilic phosphate heads exposed to the water content outside and within the cell, and the hydrophobic tails hidden on the inside. In general, small, non-charged molecules such as oxygen or carbon dioxide can freely cross the membrane without an input of energy. They're able to slip between the heads of the phospholipids and pass through the hydrophobic tails through a process known as diffusion. These molecules are able to move down their concentration gradient as they move from an area of high concentration to an area of lower concentration. In contrast, ions and polar molecules have difficulty crossing a membrane. To move as quickly as is necessary, they are often assisted across the plasma membrane by carrier proteins. The proteins that conduct this form of transfer are often called pumps because they use energy to force molecules or ions to move from an area of lower concentration to an area of higher concentration. This is commonly referred to as up or against the concentration gradient.
So, section 3.3, passive transport mechanism, passive membrane transport. So, remember, passive transport requires no energy input, so no ATP required for movement of substances. There are three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. So, when we say the word diffusion, we mean that it's a natural movement of molecules from areas of high concentration of that molecule to areas of low concentration. And this we also refer to as moving down a concentration gradient. Ions and molecules are constantly in motion, colliding and bouncing off one another and off obstacles in their paths. The movement is random, so a molecule can bounce in any direction. One result of this continuous random motion is that over time, the molecules in any given space will tend to become evenly distributed. And so this distribution process is called diffusion. As the molecules move around, there will be a net movement of materials from areas of higher concentration to areas of lower concentration. And the difference between the high and low concentrations is a concentration gradient. Diffusion tends to eliminate that gradient. Figure 3.5 illustrates the concept of diffusion. So let's say you put in a dye pellet in water, for example. So there's a concentration gradient at the beginning. So it's a difference in concentration. You have an area of low concentration of dye, at time 0, for example, and an area of high concentration of dye. Then over time, the random thermal motion, kinetic energy, makes the dye molecules move and collide against each other constantly. And as a result, they move down their concentration gradient from high to low. And when the dye is evenly distributed, then there's no more net movement of dye because there's no more concentration gradient.
Diffusion is the net movement of a substance from a region of high concentration to a region of low concentration. Let's use this sugar cube and beaker of water as an example. When the sugar cube is dropped into water, it begins to dissolve. The sugar molecules in the cube move from an area of high concentration to areas of lower concentration until the molecules are evenly distributed. This type of motion is known as diffusion.
So the speed of diffusion is influenced by three factors: concentration, molecular size, and temperature. For concentration, the greater the difference of concentration between two areas, the faster the diffusion occurs. For molecular size, smaller molecules will diffuse faster than larger molecules. Temperature, higher temperatures will increase the kinetic energy, which results in faster diffusion. And equilibrium is reached when there's no net movement of molecules in one direction only. Molecules have a natural drive to diffuse down the concentration gradient that exists between the extracellular and intracellular areas. And we can see these examples, next semester for sure, when we look at nerve physiology and how sodium and potassium follow their concentration gradients. But nonpolar hydrophobic lipid core of the plasma membranes stop diffusion and create concentration gradients by acting as selectively permeable barriers, which is also called differentially permeable barrier. There are molecules that are able to passively diffuse through the membrane without the help of special carriers or transport proteins, and these include lipid-soluble and non-polar substances, something like, for example, cholesterol. Also, very small molecules that can pass through the membrane or membrane channels. And this is referred to as simple diffusion. So things like water, CO2, oxygen, these can pass through. Larger or non-lipid soluble or polar molecules can cross the membrane, but only with assistance of carrier molecules. And this is referred to as facilitated diffusion because they can't pass through on their own. So things like glucose would need a carrier molecule to facilitate its diffusion. Osmosis is a special name for movement of solvent, usually water, and not molecules. So it's a special type of diffusion, but it's the diffusion of water from an area of high concentration of the water to low concentration of water. Clinical homeostatic imbalance 3.2. If the plasma membrane is severely damaged, substances diffuse freely into and out of the cell, compromising the concentration gradient. A good example of that is seen in burn patients that lose precious fluids, proteins, and ions that weep from damaged cells. So for simple diffusion, nonpolar lipid-soluble hydrophobic substances diffuse directly through the phospholipid bilayer. Examples are oxygen, carbon dioxide, steroid hormones like cholesterol or estrogen, and fatty acids. Small amounts of very small polar substances such as water can even pass through. Figure 3.6a, diffusion through the plasma membrane through simple diffusion. Here you can see the extracellular fluid that separated from the cytoplasm by the lipid bilayer, and you have a lipid-soluble solute that is moving from an area of high concentration to an area of low concentration through the phospholipid bilayer without any help of carrier molecules. In facilitated diffusion, certain hydrophobic molecules like glucose, amino acids, and ions like sodium and potassium are transported passively down their concentration gradient either by carrier-mediated facilitated diffusion, where substances bind to a protein carrier, or through channel-mediated facilitated diffusion, where substances move through water-filled channels. In carrier-mediated facilitated diffusion, the carriers are transmembrane integral proteins. Each carrier transports specific polar molecules such as sugars and amino acids that are too large for membrane channels. Binding of the molecule causes the carrier to envelop it and change shape, that results in the molecule being moved across the membrane in the process. Binding is limited by the number of carriers present, so carriers are saturated when all are bound to molecules and are busy transporting. 3.6b shows diffusion through the plasma membrane via carrier-mediated facilitated diffusion. So here we have a lipid-insoluble solute such as a sugar or an amino acid that is going to, via a protein carrier specific for that chemical, is going to bind, and the transport protein is going to change shape and allow the movement of that solute across the membrane, again, always from an higher concentration of that solute to a lower concentration of that solute. The other example of facilitated diffusion is channel-mediated. So these channels with aqueous-filled cores are formed by transmembrane proteins. The channels transport molecules such as ions or water down their concentration gradient. So specificity is based on pore size and or charge, and the water channels are specifically called aquaporins. We're going to see examples of this type of transport when we look at kidney physiology next semester. There are two types of channels: leakage channels, these are always open, and gated channels. These are controlled by chemical or electrical signals. Figure 3.6c, diffusion through the plasma membrane via a channel-mediated facilitated diffusion. So these small lipids insoluble solutes will pass through a channel protein, and it's mostly ions, and they're selected on the basis of size and charge. So positively charged sodium, for example, will pass through. Again, always from an area of higher concentration to an area of lower concentration because we're dealing with diffusion. So osmosis is another passive process of movement, but here it's the movement of a solvent, not molecules. So it's usually water across a selectively permeable membrane. Water diffuses across the plasma membrane through the lipid bilayer. Even though water is polar, it is so small that some molecules can sneak past the nonpolar phospholipid tails or through specific water channels called aquaporins. And the flow occurs when water or other solvent, when the water concentration is different on the two sides of a membrane. In other words, where one side it would be higher compared to the other side. Figure 3.6d, diffusion through the plasma membrane, specifically osmosis, which is the diffusion of water molecules. And this could happen through a specific channel protein called an aquaporin or through the lipid bilayer. Osmolarity is measures the concentration of the total number of solute particles in solvent. Water concentration varies with the number of solute particles because solute particles will displace the water molecules. So when the solute concentration goes up, the water concentration goes down and vice versa. So water moves by osmosis from areas of low solute or high water concentration to high areas of solute or low water concentration. When solutions of different osmolarities are separated by a membrane permeable to all molecules, diffusion of solutes and osmosis of water occur cross membrane until the equilibrium of solutes and water is reached. Occur across the membrane, equilibrium is when you have the same concentration of solutes and water molecules on both sides with equal volume on both sides. Figure 3.7a, influence of membrane permeability on diffusion and osmosis. So in this first slide, please note the membrane is permeable to both solutes and water. So the solution with the lower osmolarity on the left, and you have a solution with higher osmolarity or higher solute concentration on the right. So over time, if both solute and water molecules can move down their concentration gradients, they will reach equilibrium and the two solutions will have the same osmolarity and the volume is going to be unchanged. In another scenario, when solutions of different osmolarities are separated by a membrane that is permeable only to water, only osmosis will occur until equilibrium is reached. So water will have net movement across the membrane until osmolarity is the same on both sides, and this will result in a volume change on both sides. The low solute side volume will decrease, whereas the high solute side volume will increase. Figure 3.7b illustrates this scenario. So please note the membrane is permeable to water, but it is impermeable to solutes. So water, but not the solute, can move through the membrane from an area of high water concentration to an area of low water concentration, or an area of low solute concentration to an area of high solute concentration. And when water moves by osmosis from an area of higher to lower water concentration, then at equilibrium, both sides have the same osmolarity. However, the volume will be different because, remember, only water can move. The movement of water involves hydrostatic pressure and osmotic pressure. Hydrostatic pressure is the outward pressure exerted on the cell side of the membrane caused by increases in volume of the cell due to osmosis. This is also referred to as the back pressure. The osmotic pressure is the inward pressure due to the tendency of water to be pulled into a cell with higher osmolarities. So the more solutes inside a cell, the bigger the pull on water to enter, resulting in higher osmotic pressures inside the cell. When the hydrostatic pressure equals the osmotic pressure, there's no further net movement of water, which means that water will move, but there will be no net movement. So water trying to get out equals water trying to get in. Now, in plant cells, because they're surrounded by a strong cell wall, it acts to limit the hydrostatic pressure levels, which in turn limit the osmotic pressure. So plant cell will fill with only so much water, then it's going to stop, which is a good thing because, even after periods of long rain, plant cells are not going to burst. Animal cells, however, do not have a cell wall, therefore they cannot limit the hydrostatic and osmotic pressures, and animal cells will burst if too much water is taken in. Also important to know is that water can leave a cell, causing it to shrink, and changes in cell volume can disrupt cell function. Tonicity is the ability of a solution to change the shape or tone of cells by altering the cell's internal water volume. So there are three solutions: isotonic, hypertonic, and hypotonic. In a isotonic solution, an isotonic solution has the same osmolarity as inside the cell, so the volume remains unchanged. In a hypertonic solution, it has a higher osmolarity than the inside cell, so the water flows out of the cell, resulting in cell shrinkage. Shrinking is referred to as crenation. In a hypotonic solution, this one here has a lower osmolarity than inside the cell, so water flows into the cell, resulting in cell swelling, and this can lead to the cell bursting, referred to as lysing. The osmolarity is equal to molarity times the number of ions or particles. For example, sodium chloride is one particle, but it ionizes to sodium and chloride, which are two particles. Therefore, 1 molar solution of sodium chloride equals to 2 osmolarity solution. So the osmolarity is expressed in osmoles per liter. Figure 3.8 shows the effect of solutions of varying tonicities on living red blood cells. So this is a practical example of how tonicity works. So in figure A, in an isotonic solution, by the way, iso means same. So that means that the concentration of the red blood cell is the same as the surrounding solution. So cells will retain their normal size and shape in an isotonic solution. So water will move in and out, but there's no net movement of water. In a hypertonic solution, seen in part B, the cells, because the surrounding solution is more concentrated, the cells will lose the water by osmosis and shrink because the solution is hypertonic. Hypertonic means that it's more concentrated compared to the inside the cell. It means that there are more solutes outside than inside the cell. So water is going to move from an area of high water to an area of low water, right? So high water inside the red blood cell to an area of low water outside. In a hypotonic solution, that means hypo means less concentrated solution, the outside of the cell is less concentrated than the inside of the cell. So that means the inside is hypertonic and the outside is hypotonic. So cells take on water by osmosis until they become bloated and burst or lyse in a hypotonic solution, which contains a lower concentration of non-penetrating solutes than are present inside the cell.
Before we take a look at how osmosis and tonicity affect a cell, let's review what each of these terms means. Osmosis represents the diffusion of water across a semi-permeable membrane. The term tonicity refers to the relative solute concentration of two environments separated by a semi-permeable membrane. In other words, By comparing the tonicity of the solutions, you can determine the direction in which osmosis will occur. To demonstrate how tonicity affects a cell, let's place some red blood cells into a beaker containing pure water. In this case, the solute concentration is greater inside the cells than in the surrounding water. In other words, the contents of the cells are hypertonic in relation to the hypotonic contents of the beaker. Because of this, osmotic pressure results in the diffusion of water across the membrane and into the cells. Over time, if enough water enters the cells, the cell membranes may burst. This is called lysis. Now let's place the red blood cells in a beaker containing a solution of salt, such as sodium chloride. Since the contents of the beaker are hypertonic in relation to the interior of the cells, the water within the cells will diffuse across the membrane and into the contents of the beaker. This causes crenation, or shrinking, of the cells. If the red blood cells are placed in a beaker whose contents match the tonicity within the cells, then there is no net gain or loss of water. The environments within the beaker and inside the cells are said to be isotonic, or the same. An important thing to remember is that osmotic pressure always causes water to move from a hypotonic environment toward a hypertonic environment. In other words, water moves toward areas of high salt or sugar concentrations. This simple process is used to drive the operation of our kidneys and explains some of the physiological consequences of diseases such as diabetes.